Welcome back to what is the last session. We're gonna run through sessions 4 and 5 together for the last part of this excellent meeting. My name's Andrew Redmond. I'm the current acting director of infectious diseases here at Royal Brisbane. We've already had a couple of acknowledgements of country, so I might just accept those as given. So look, I've got the delight in introducing our speakers today, and our speakers are really from diverse areas, occupying different roles in supporting transplant patients in a variety of ways. First up, we've got Patrick Harris. Patrick is again the acting statewide director of microbiology at Pathology Queensland, as well as being a UQ Amplify Fellow at UQ-CCR. His research has focused on antibiotic-resistant bacteria and the use of randomised controlled trials to define optimal treatment for these problematic infections, as well as the application of microbial genomics to clinical practice. Okay, thanks, Patrick.
So look, obviously I can't possibly cover all of transplant microbiology in 15 minutes. So I'm going to be just picking and choosing a few things. Really, I guess I'm sorry if there's people listening in America or somewhere more exotic. This is going to be rather parochial and talk about, I guess, things that are happening at the PQ lab here, and I guess things that may be relevant for microbiological diagnostics in these difficult patients. And as we've heard about today, there's a huge array of complexity and different types of infections we may have to encounter in these kind of patients. And this is the great challenge for microbiology, is you often don't quite know what you're looking for. I guess you can risk stratify, and we know sort of relatively predictably what sort of infections may occur in certain types of patients at certain times during their journey. But often there are many surprises. And this is the real challenge, is sort of picking the right tests and the right kind of requests to make in your samples to get the best sort of diagnostic yield. Now, as this is, I think, what most people think the microbiology lab, and in fact, in many ways, it hasn't changed an awful lot. I think Pasteur would wander in today and be quite happy. And but things have evolved a little bit, and I think hopefully we've moved on from traditional culture-based microbiology and gardening, but not maybe that far yet. So I'm going to talk about a few things that I guess have been happening in the lab that I think have relevance, particularly for these kind of patients, that it might be worth just talking about, and so that you're aware of the kind of things that you can order and use for your clinical management. So a bit of a background to, I guess, the microbial genomics journey. It's been a bit of a saga, and it's actually been going on for several years now, but some of you may know that there was this initiative through the Queensland government called initially the Queensland Genomics Health Alliance, but later rationalised to Queensland Genomics. And there was $25 million from the state government to, I guess, bring genomics into healthcare at the front line within Queensland Health. Now, as you can imagine, most of that was for things like cancer or rare genetic diseases, but we managed to carve out a little bit of money for microbial genomics. It was initially completely forgotten about, but we sort of put a hand up and said, bacteria have genomes too. And I guess we thought about what could we do in the genomic space that could be really eminently translatable, and we could sort of bring to sort of the clinical sort of laboratory quite quickly. And we thought the most useful application would be in the same way that in public health, genomics is now pretty standard for outbreak investigations of salmonella or listeria or those sort of public health notifiable diseases. We thought it'd be really relevant to try and apply this to infection control and multi-resistant organism surveillance. So we cobbled together basically the kind of whatever we could find in the environment here in Brisbane, from the university, from FSS, the forensic laboratory, what we had at Pathology Queensland. We got our bioinformatics friends at the Beatson group at UQ, people here at UQ-CCR, etc. Just pulled together all the expertise we could into this project to try and see if we throw genomics at sort of infection control surveillance, could that be useful, and could that be something we could actually ultimately apply in the lab? So the idea was basically just to take all our sort of multi-resistant organisms, like the kind of things we've been talking about today that are very problematic to treat, to sequence them prospectively. So before you identify, transmission events or clusters, or you have big outbreaks, and try and, I guess, provide that early warning that there may be clustered isolates, and you could generate reports back to infection control teams. Now, this took many years to kind of piece together, and it's been a fairly slow process to sort of integrate all those systems, but we're sort of getting somewhere. We working with the UQ Bioinformatics group, we've built this fantastic pipeline called Snapper Rocks, which essentially pulls together all the patient metadata from the medical record from OzLab and from elsewhere. So we have things like admission dates, ward locations, as well as the genomic data. And the genomic data can be fed through this pipeline straight from the instrument, and then can generate things like species, taxonomic profiling, sequence type, MLST, and ultimately things like SNP profiling. So looking at the number of mutations in comparison between different genomes. So you can start to see when isolates are very closely related, and there may be outbreaks or other transmission events of note. So I lost my thing, here we go. So this now actually, believe it or not, so we've brought OzLab, good old 1980s MS-DOS technology, can actually now report genomics results. So you will start to see these coming into OzLab. We've actually managed to get this NATA accredited. So this is, I guess, a huge amount of work, and a lot of credit to the PQ lab who've actually brought this to clinical production. And if any of you have gone through a NATA accreditation process, it is a very, very intensive thing. So there's a huge gap between research and something you can actually use for clinical use. Now, obviously, putting all that genomic information into a fairly scanty green screen report, it's missing an awful lot of the complexity. But certainly the sort of key elements are there, things like sequence typing, antibiotic resistance genes, and we sort of report, some of the key resistance genes like carbophenamases, ESBLs, and so on that we think might be clinically relevant. The other thing we've had this portal called CathEye, which I meant to have a picture, but I don't think I put it up. So CathEye is actually a web-based portal, so you can log into that, and then you can explore that data yourself, and you can start to look at different types of relationships between certain species or different sequence types. You can look at your hospital or certain wards and start to see where you may have transmission events occurring in your hospital. So I think, hopefully those of you particularly working in infection control, infectious diseases, will start to see this. And if you want to use it more, let us know and we can sort of show you how to do it. But that's for infection control. I guess, as I said, this was our sort of Trojan horse to sort of get this sort of genomic facilities up and running within the laboratory. But once you can do sequencing, you can then start to apply it in all kinds of different ways. This is a nice example. We were just talking about Elizabeth Kingia a minute ago. This is a nice paper that Adam published a few weeks ago. This was another very similar case of an immune-compromised patient who had a persistent Elizabeth Kingia bloodstream infection, also had meningitis. We tried throwing all kinds of different antibiotics at it, and again, rather sequentially, and this is again where we've sort of been thinking more about the need for combination therapy, because every time we threw an antibiotic at it, you get resistance developing. So here you can see resistance to rifampicin and ciprofloxacin developing. And we were able to sequence this and quite quickly show that, you had associated mutations to account for that resistance. Now, again, I think more and more we may be able to start applying these kind of genomic testing to our patients, perhaps even preemptively before we can sort of maybe even to detect silent mutations that may become apparent later on through testing. I think we haven't even begun to explore the possibilities. But the good thing is we now have that available in the lab. So you can phone me up and say, please do whole genome sequencing on isolate XYZ. It'll cost you $400 a pop. But that goes back to the hospital. It gets lost in the pathology budget. I don't think anybody even notices. So one eventually. So we'll see how it goes. And the great thing is the more of the sequencing we do, we can then start putting it on higher throughput instruments. It'll become much, much cheaper. At the moment when you're only doing 3 or 4 a week, it becomes quite expensive. So let's see how we go and wait for somebody to stop us. But I think there's a real huge possibility here. I guess the golden dream of sort of genomic-based diagnostics is sort of metagenomics, and I think that's definitely. We've heard a little bit about this earlier with sort of cell-free microbial DNA and carriers testing and so on. But the dream would be, in theory, you can take any biological specimen, any clinical sample from a patient with an infection, sequence all the nucleic acids in that specimen, and then potentially pull out potential pathogens and their associated AMR genes or virulence or whatever to make a clinical diagnosis. Now, obviously, that's a very simplistic way of looking at it. It's way more complicated. It's actually incredibly challenging. But I think this is definitely where microbiology needs to go. And certainly there's a huge amount of interest and work going on in that direction. So we've actually been looking at a few things. There are some now commercial platforms coming. So Illumina, as you may know, is, I guess, the sort of very standard sort of Rolls-Royce, not Rolls-Royce, it's more like a sort of Nissan Micra. Everybody has one, the sort of standard, reliable sequencing platform that are available in most laboratories now. But they're bringing out these enrichment panels. So rather than having to sequence all the DNA, which is relatively wasteful, most of it's human DNA, the pathogens might be there like a needle in a haystack, and sometimes you're not even sure if there even is a needle in that haystack. So a lot of background contaminated DNA, so pretty complex material to work with. The idea is here is that you actually amplify up a panel of pathogens, rather like a massive multiplex PCR. So you might have, potentially hundreds of targets, even thousands of targets. You amplify them up and then sequence all those amplicons. There's ways it can be hybrid capture, it can be amplicon based, but essentially there's sort of enrichment prior to sequencing to improve your sensitivity and specificity. And there's a panel like this. This is the RPIP. This is the respiratory-based panel, which has over 280 respiratory pathogens, including viruses, fungi, bacteria, mycobacteria. In theory, anything that you might want to consider in a complex immunocompromised patients with a lung infiltrate or something like that. And also having over 1,000 AMR genes as well, so potentially also guiding therapy. And they're really sophisticated now, so they actually will have a bioinformatics analysis attached to these panels. It sort of goes up into the cloud somewhere and will spit back a report, which looks, a bit like this, is the sort of thing that we're relatively familiar with for a clinical report, where it gives species that's identified. It gives you sort of bacterial, at least DNA loads. It'll give you some sort of interpretation of resistance markers and what those resistance markers might mean in terms of treatment. Here you've got bacteria, you've got viruses. Again, you can argue about the significance of all these things and how are we going to interpret this. But I think it's really interesting to see that this stuff is coming very, very quickly. I think we're going to have to do a huge amount of work to validate this for clinical use. It's extremely complex. We're going to get a lot of background noise. You pick up CMVs and EBVs and all kinds of things, as we do at the moment doing singleplex PCRs. But I think we're going to have to learn more and more how to interpret this and use it clinically. But I think it's pretty exciting. So that's genomics, and I think that's got all kinds of great potential for the lab, and I think we're sort of making our first inroads into that direction. Some slightly more mundane but really important things that we're doing is trying to really get a better handle on our fungal diagnostics. I think the reality is, as we've heard today, fungal infections are incredibly important, becoming much more complex and common in our patient populations. We previously had to send away most of our fungal testing, particularly for sort of susceptibility testing for molds, or if we had difficult organisms we couldn't identify. Certainly things like molecular diagnostics for Aspergillus PCR or panfungal PCRs all would have to go down to New South Wales or Adelaide. Galactomannan we actually had to send to Sullivan Nicolaides. The turnaround time was, pretty awful. No offense to Sullivan Nicolaides, that's the way it is. So I think we sort of bitten the bullet, so we really have to try and bring as much of this in-house. So I think you'll certainly see over the coming months and years that a lot of this will start to hopefully be offered at PQ ourselves. We now have a MALDI-TOF for mold identification kit, so that's really sped up and made identification of these tricky molds much, much faster and more reliable. It's not yet applicable to everything, but it's pretty good for your common molds. Little things like a suvaconazole MIC testing, and we're, again, trying to expand our repertoire of the kind of MICs we have available in-house. And I think the big thing is going to be validation of an in-house mold MIC capability at PQ. Now, again, mold susceptibility testing is a whole dark art in itself, so it's not something you can just do straight away. They're also very hard to interpret. There's often no clinical breakpoints. But I think until we start really doing it, getting a handle on it, generating our own local data and experience, that's the only way we're really going to proceed and make some progress. And then I think importantly for diagnostics, we are working on an Aspergillus PCR. As we heard earlier, there are some commercial offerings that are coming onto the market. So we're looking at a few of those, and hopefully we'll have that underway before too long for your patients. And then there are other things, like this morning we saw that patient that had HHV6 encephalitis. So actually that was one of our early BioFire ME panels. So that's meningoencephalitis panel. This is something we've been trying to do for a long time. You may know, for these more extended multiplex panels, we had to send them away to the Mater Hospital, particularly in neonatal meningitis, but also very relevant for other immune-compromised patients, for Listeria, HHV6, and so on. But now we've replaced that with the BioFire in-house. But just be aware that, again, it's pretty expensive. It's about $400 a test. So there's a little bit of restriction in terms of who we would suggest it's used on, but certainly for these kind of complex patients, it may well be appropriate, particularly if your first-line PCRs are all negative. So those are the kind of targets that we can pick up. And as we said, it certainly covers the vast majority of your most common organisms, including some of the slightly more esoteric ones. It's very quick. It's very easy to use. The lab staff love it because it's almost minimal hands-on time. Just a few little caveats. Just be aware there have been some case reports of false negative herpes 1. So certainly if you have a very high index of suspicion for herpes encephalitis, particularly MRI changes or other sort of classical features, you may want to back that up with an in-house PCR. We haven't found this in our validation data, I have to say, but it has been reported elsewhere in the world. And I think the other key thing to remember, particularly for the kind of patients we're talking about today, it doesn't cover the really weird and wonderful stuff. You're not going to get your toxo, your JC virus, your mycobacteria, etc. So it still requires, unfortunately, a bit of diagnostic thought. And another thing to remember is, although there is a cryptococcal target on the PCR, it's not as good as the antigen. The antigen still remains actually a much better test. So the last thing I'm going to just finish up with, sorry if I'm taking a bit of time, but the IDL is this lab we built actually during COVID. It's called the Infectious Disease Laboratory. It's actually based over at QIMR. We built that primarily for sort of mass respiratory diagnostics. So they have lots of high throughput molecular instrumentation and robotics and can do all kinds of interesting things. Obviously, the COVID testing has gone off a cliff. We've kind of been told we have to shut it down and chuck everything in the bin, which I think is a terrible idea. So we're trying to rebadge it as a kind of research and development space for PQ. So anybody who has asked PQ in the past, can you develop this test for us or something else? As you know, it's a pretty painful experience. I see Julia nodding. So the Kingella, when I came here about 10 years ago, I remember we were talking about Kingella PCR, and it's sort of been grumbling in the background, and we've sort of never quite got it over the line. So Ian Mackay and his team at the IDL probably got it over the line in about 2 months. And now it's orderable in OzLab, and it's validated for clinical use, within a period of months. We've also brought the Coxiella PCR, the Q fever PCR that used to be done at FSS, relatively slow, not many people weren't even aware that it was available. That's now done at the IDL with same-day turnaround time. And in fact, we've been adding PCR requests to all our negative serologies from high-risk patients, and we've got about a 12% pickup rate for PCR. So it's a real lesson that we're probably missing a huge amount of Q fever just by serology alone. And they're actually brilliantly set up now to develop new assays. So in response to the Japanese encephalitis outbreak or the monkeypox outbreak, they were able to develop in-house PCRs really quickly to respond to that. They're looking at things like quantitative HHV6, which, as we heard, can be potentially quite useful for those sort of complex patients. They're also looking at a kind of North Queensland unexplained fever assays for Brucella, Rickettsia, Lepto. And if you could combine that into a multiplex PCR, that could be really useful. So the way I'd like to think about the IDL is a potential sort of research space for assay development that's useful for you. So, if you guys come to us and say, we really need a test for XYZ, we can certainly consider doing that. And the more we use the IDL, the more we show that it has this worth to the system, it makes it harder for them to shut us down. So on that positive note, I'll stop there, and hopefully I haven't gone too far over time. Thank you.
Right. So look, Patrick was very efficient and we do have time for one or two questions. Please, Antia.
Thoughts on technologies like IgA, where you can pull down bacteria which are reactive to IgA and sequence that to sort of pull out more immunoregulatory bacteria or things that are probably a little more informative than just sequencing every bug that's there, just like a pull down, if you like.
Yeah, I mean, look, and this is what's so exciting. There's so much happening, I think, in the research space, and there are all these exciting things happening. The biggest problem is there's such a huge gulf between that and bringing that into clinical production. So something that you can present to, and the regulatory environment, we can't just do research use only tests and report them anymore. You can, but the sort of the infrastructure around that and the rules around that are becoming more and more complicated. So you have to be very, very careful in what you're doing. And I think even, for instance, when we went through our genomic validation with NATA, I don't think even they quite knew how to validate it. There's not many that haven't done this very often for diagnostic labs. So everybody's sort of feeling their way. I think we'll have to start with the more straightforward things, things like characterizing a cultured organism and looking at for AMR genes or something. That's fairly straightforward and you can validate it against other fairly well-established assays. When it comes to slightly more cutting edge things, I think there's inevitably going to be quite a long period before it really makes it into clinical production, I think, in reality, particularly when we don't always know 100% what it really means or how clinicians would use that information. But it only comes through experience and, trying. But it's not easy. And I think there's always going to be that sort of lag, unfortunately.
Patrick, does your genomic data output include a direct analysis to determine whether or not the organism has been found previously? So do you keep a historical data set, database, so you can see if a
we've now got
problem strain turns up again?
I think we're nearly sort of 4,000 or 5,000 genomes in the database now. So, and that's the great power of it. It makes it more computationally complicated. And then some of these clusters like our ST131 is like enormous and it's getting a bit of a jumble. But that's the great thing. We can very quickly say, okay, that strain, that's 3 SNPs apart from an E. coli we picked up in a toilet in, Prince Charles. And actually, it's been really helpful to just actually, rather than everybody guessing as to what's going on, you can actually point them pretty quickly in the right direction. Now, it depends how much you're sampling. And the great thing was that initial project, because it was funded through the government and they gave us quite a bit of money, we could do a lot of sequencing upfront. But so the sort of things that we wouldn't normally have sequenced probably, but we now have this incredible sort of data set that we can compare to.
Great.
Just one more question from Crispin on metagenomics.
It's actually about the cost, and I guess it's a good comment and a question about making sure we do evaluate the cost of this technology versus existing technology. The ID unit just had a patient through with a fever of unknown origin where I counted up that we performed 60 separate,
exactly.
microbiological, serological, and molecular tests and a PET scan and a CT and an MRI, I think. And, by the time you're starting to do that, some of this starts looking quite cost effective. And I'm just commenting that it's probably really important that we could probably show.
I mean, that RPIP kit, each kit costs about $200, maybe additional $100 for consumables and labor or something, a bit, maybe let's say it's $400. By the time you add up all the PCR, I mean, all these patients get respiratory full panel and a 4-plex gene expert when they come in the door. And they get serology and they get AFBs and they get fungi and they get galactomanans and a PJP and
they get a whole lot of non-infection workup as well. They get anti-nuclear antibodies and all this sort of stuff as well. And it must cost thousands. And they also stay in hospital for 2 or 3 weeks while that's happening as well.
Yeah, exactly. So, I think if we can show, particularly from a pathology perspective, yes, there's savings to the health system, which is always a bit harder to show in the pathology world. But if we can potentially replace a lot of particularly very labor-intensive microbiology, like TB diagnostics. A lot of places now, they go straight up to, they don't even do TB susceptibility testing. If they find no resistance genes, they'll just report it out as fully susceptible TB. And, 6 weeks waiting for AST for TB is kind of hopeless, really. Now, you're probably still going to need it for your more complex, more resistant isolates and so on. But if For your bog standard wild type TB, there's probably no point. So I think we're going to have to change the way we do things. But if we can save labour and other things, that's going to make it justifiable, I think.
Wonderful. Thank you so much.